Cancer, it's a very bleak topic, but it is a very important one, and typically one that medics tend to shy away from, especially in first year, the dreaded cancer week. But it is, like I said, very essential, so do try and, stay with it. This is going to be more of a basic introduction into what cancer really is. So let's get started. Now, we do unfortunately have to start with some, basic definitions before we get into the nitty-gritty. So ignore the first one for now, because the actual definition of cancer we'll talk about in a second. Let's start with carcinogenesis. So cancer doesn't just happen randomly. It takes time. It's an ongoing process. So it's multi-step, and it involves the transformation of a normal cell to a cancerous cell type. And that doesn't happen easily. So we're going to talk about the details of what goes on to cause cancer later on. Carcinogens are compounds such as, chemicals, radiation, or microbial, because you'd be surprised at how many cancers are caused by infections. And these induce cellular changes. They damage the cells, they damage the DNA that eventually develops into cancer. Differentiation, that's another key one. It's usually a normal process of development. The maturation of a cell, allowing it to perform a specific function. As for cancer itself, now, please do note this is a very basic definition. The real thing includes the hallmarks, which we'll be talking about in a second. But essentially, it's characterized by unregulated growth of malignant tumors. Now, malignant refers to the next part, the potential to metastasize and spread to other regions. So these are not the kind of tumors we want. We don't want any, but benign tumors, they don't really spread to other parts. They're not as problematic. So let's talk about the hallmarks. By the way, in terms of what you actually need to know, the stuff on the slides, it's more than enough to give you the basic understanding. I would advise you look into some statistics about the most common cancers and things like that, but everything else is pretty much covered to a decent level of detail in these slides. So use these as a basic foundation, and we'll talk more about cancer as we go along. This is going to be the first of a relatively long series, because it is quite an in-depth topic, and there is a whole week dedicated to at least in Barts for medicine in first year. So hallmarks, now these are the things that kind of define cancer. There are a couple of others, but the basic 5 that you need to be aware of are these. Let's start with evasion of apoptosis. So apoptosis is the process of programmed cell death. Programmed is a very important word here. Cell death occurs all the time, right? We're always damaging cells and stuff's going wrong. But you have to control it, because when cells die, they release a lot of harmful compounds into their environment, and that can damage other cells and cause even further death. So apoptosis is basically when the cells in our body realize, oh, hold on, something's gone wrong with me. Either they essentially kill themselves, or their neighbors kind of gang up on them and kill them. And as brutal as it sounds, it's actually very, pre-planned, premeditated, and well designed to basically clean all the damage up. So they destroy that one cell that's damaged, and then they clean up all the mess and make sure it doesn't spread anywhere else. Now, cancer is able to evade apoptosis, so it's able to survive that programmed cell death, so it doesn't die as easily. So that's a crucial thing here, because remember, cancer is basically, you can think of cancer as like a hyper-evolved organism. It's almost got a life of it, well, it does have a life of its own, but it's almost as if it's got a stronger will to survive than other cells, because it's relentless. That's why cancer is so difficult to treat, because it's primed for survival. If you think about it, it's viciously proactive in terms of what it needs to do to live. And it occurs partly because of DNA repair defects. So this is another hallmark. DNA is always being replicated in our bodies, right? It's how growth and repair occur. Now, things go wrong all the time because there's so many cycles and processes going on inside the body. Things are bound to go wrong. And that's why literally every single organism that has more than one cell is known to have cancer, or suspected to have had cancer at some point, okay? It's kind of an inevitability of life. Obviously, that's not saying that everyone's going to get cancer, but most multicellular organisms make them live long enough, and they will at some point probably develop a cancer. Just because of how much DNA repair and replication there goes on inside the body. Every now and then, however, defects occur. There's loads of these, and we're able to take care of most of them. We have, safeguards and around us that protect our cells really and stop cancerous cells from growing. But sometimes these go wrong. Sometimes these safeguards fail, and we'll talk about some of them and what they are and what they do in a second. But when they do fail, you do get these repair defects that can eventually, again, it's multi-step, so it's not like one mutation or one defect is immediately going to give you cancer. It's usually a combination of a few different defects or genes or proteins combined with some environmental stimulus, and that eventually leads to this cancer, which is also characterized by its invasive metastatic potential. So, like I said, the crucial thing separating benign and malignant tumors are malignant ones want to spread. Like I said, they've got this brutal kind of, strive to keep living, and they will invade other parts and just try and take over like an army, really. They're able to do this because they've got replicative immortality. What that means is cancer cells theoretically can replicate forever. Now, normal cells, as they replicate, the chromosomes get shorter and shorter. I'm not going to talk about the in-depth detail here, but they get shorter and shorter until eventually they're so small that the cell kind of just dies. And that is called the Hayflick limit. That's a limit to the number of times a cell can divide. Somehow, we don't fully understand the mechanism, but somehow cancer cells are able to avoid that. So they're able to keep lengthening the chromosomes so they can technically replicate forever. And that is what really kind of causes cancerous cells to grow so quickly and rapidly with no end in sight. And then finally, angiogenesis. Again, like I said, cancer wants to live. It's its own life, really. So in order to do that, it creates its own blood vessels, and this is called angiogenesis. Genesis meaning creation, angio referring to blood or blood vessels. So over time, cancer cells will develop their own blood supply to basically survive for as long as they can and kind of steal nutrition from the body. Now, let's get into the genetics, kind of. So there's a few things we need to know here. First of all, of course, cancer can be hereditary, okay? A lot of the genes that do eventually lead to cancer, when combined with the environment, they can be inherited. But most cancer is caused by spontaneous mutation, and this surprises people, but it is the case. So most things occur because of environmental stimuli combined with perhaps, lack of nutrition, lifestyle factors. Think about it, like that's why smoking is such a big deal for lung cancer. Of course you can inherit cancer of the lungs, but the main crucial thing is the damage from the tobacco and the tar and the smoke and the chemicals in there. And those lead to spontaneous mutations by damaging DNA and proteins. So that is the most common cause of, cancer. That's really where it tends to come from. But there are, a few kind of genetic things that we do need to know about cancer. So we've got basically 4 different categories here of different proteins and genes that can either be inherited or developed over time. That link in with cancer. So the first is an oncogene. Onco refers to cancer, gene as well. We know what gene is. It's a section of DNA that codes for a protein. Remember A-level biology. So an oncogene basically causes cancer. Think of it like a criminal running around on the streets causing damage, okay? We do not like oncogenes. We want to down-regulate them, keep them as low as possible. Proto-oncogenes are pre-derivatives of oncogenes. So these are genes that they're not necessarily inherently bad, but if something goes wrong, they can quickly turn into oncogenes. To think of these as like high-risk categories that can turn into criminals in real life, people in deprived populations, for example, okay? So these usually regulate cell proliferation, differentiation, and mutations. They kind of, take care of all of that stuff, slow them down, stop them happening, etc. When you get mutations or downregulations or even upregulations of certain aspects of these proto-oncogenes, they enhance the signaling pathway. We'll talk about what that means in depth in a bit, but that can essentially mean the growth signals are increased. So basically your body's saying to these cancers, keep growing, keep growing, keep growing, okay? Or in fact, not the body, it's more the cancer tells itself to grow. So it basically, one cancer cell communicates to this cancer cell next to it and says, we have to keep going, don't stop, don't stop. And that's what eventually leads to cancer. We've also got ones called tumor suppressor genes. Now these are like our gatekeepers. They establish balance, control, and apoptosis in the body. Think of them as like courts, the courts of law, okay? So they are the ones that sentence cells that have become damaged to death. via apoptosis, so programmed controlled cell death, and they basically maintain a neutral environment inside the body, as far as DNA damage is concerned anyway. You've also got some repair proteins. Now these are like the police, okay? They kind of go around, they fix the damage as it happens before things escalate beyond repair, okay? But what happens when these things start going wrong? So let's start with proto and oncogenes, okay? So you've got 5 different categories here, okay? An example of the first one is a growth factor, okay? So growth factors, as I mentioned earlier, are things that stimulate cells to grow. An example of this is one called TGF-alpha. So if TGF-alpha is released into the blood, it goes and binds to a cell, and it says to the cell, I want you to grow, you're not growing enough, grow more, okay? These are overexpressed in cancer. They're not mutated, bear that in mind. At the same time, you've got the receptors of these growth factors, because there's no use having extra growth factors if they can't bind to anything, right? So you might have receptors which are increased as well, also overexpressed, not mutated. A key example of this is HER2. Now that's a very important one for breast cancer that as you go through your years of medicine, you'll become more familiar with, especially in 3rd year. And then, of course, you have signal transduction proteins. These ones, they're very important, but they're kind of hard to wrap your head around. So think back to biology, and also you'll cover this in other kind of aspects of the fundamentals of medicine in 1st year. But think of how, you know, protein signaling occurs with like GLP proteins and all of that stuff. So basically you've got GTP, which is a compound that binds to your cells and stimulates signals, okay? So it releases signals. Now, in order for that signaling pathway to stop, you have to break down and hydrolyze that GTP. That hydrolysis is done by proteins that are known as RAS, okay? RAS, RAS. If they mutated, they become something called KRAS, and this is bad. This is a very hard protein to treat. Until recently, we didn't have anything for it really, but recently they've released a drug that can somehow penetrate KRAS. But anyway, that's besides the point. So essentially KRAS doesn't do the job properly. It doesn't break down GTP fast enough. Because it doesn't break down GTP fast enough, the signals continue, okay? The growth signals, they don't stop, and so the cancer reproduces very quickly. It grows more, okay? You've also got transcription and translation occurring in the body all the time, right? DNA replication, protein production, etc. So you have transcription factors that regulate transcription. So they bind to the areas of the gene that are being transcribed, and they can stop transcription occurring, or they can increase transcription, okay? If they're mutated or overexpressed, so if the ones that increase transcription are overexpressed, or the ones that downregulate transcription are mutated, they cause more transcription, and that leads to more growth, more production of proteins, and these proteins potentially can lead to cancer, okay? So that's how transcription factors work. Now, anti- and pro-apoptotic proteins, it's kind of hard to say, a bit of a mouthful. BCL2, now this is anti-apoptotic, so this prevents apoptosis. So if a cell is releasing BCL2, the body will not let it die, because it will sense the signal and it will think, oh, this cell wants to live, I'll let it live. Okay, so in cancer, BCL2 can be upregulated. BAX, on the other hand, is a pro-apoptic protein, and it's often downregulated. So if a cell releases BAX, it's basically saying, please kill me, in the nicest way possible. Okay, in cancer, that can be going down, because like I said, the cancer doesn't want to die, so it prevents BAX being released. Tumor suppressor genes. So these normally block development of cancer, okay? So if you now have a mutation of these, they lose their function. And if they lose their function, they can't block the development of cancer because that's their function. Now, 2 crucial examples of these are P53 and PRB. So P53, basically, if you get any hypoxia, which is low oxygen in the blood because that causes damage from things like free radicals, or if you get cell injury or DNA damage, P53 basically either says, okay, we need to repair this cell or piece of DNA, or we need to, just kill it. So that's kind of what it does. And if it mutates, it can't do that. PRB, on the other hand, halts progression. Sorry, there's a bit of a typo there, but it halts progression. So it basically says, oh, DNA damage has occurred, I'm going to stop the cycle of DNA replication here, and just leave it at that and let the cell die. So that one, the tumor suppressor genes are kind of simple to wrap our heads around. They suppress tumors, and if they are broken by mutations, they can't suppress tumors. What about repair proteins and genes? Now, these are ones that you may be more familiar with, even though you might not know what they really are. BRCA1 and BRCA2. Angelina Jolie comes to mind. She's a celebrity who had, I can't remember which of the two mutations it was, but it was one of them. Essentially, BRCA is a protein created by a gene that repairs DNA. It fixes issues that come up in the normal cycle of DNA replication. If you've got mutations to it, and the two key mutations are BRCA1 and 2, you have less repair, and that predisposes you to cancer, so between like 50 to 80% higher risk of cancer, depending on whether you have BRCA1 or BRCA2. These are, they're very, very common in familial breast cancer, so if you've got a family history of breast cancer, chances are it's because of a BRCA mutation. So yeah, essentially they fix DNA, and changes to them mean the DNA is not fixed, and that can lead to cancer. And really, that's kind of it. Sorry, this is a bit of a chunky lecture, despite it being somewhat of a basic introduction. Like I said, there is plenty more to come because cancer is quite a hefty topic, but it is a very interesting one once you kind of wrap your head around it. So yeah, thank you for listening. Hope it helped.